students. Today we are going to study the diagnosis and control technology of plant nematodes. Nematodes are a group of low-level invertebrates belonging to the animal kingdom Nematoda. They are called phytopathogenic nematodes or plant parasitic nematodes. When plants are affected by nematodes, the symptoms are similar to those of common diseases and are therefore often called nematodes. General traits of phytopathogenic nematodes. 1. Morphological structure. Plant nematodes are generally small, 3 to 4 millimeters long, with elongated bodies, some fusiform, and some females expand to lemon or pear shape when mature. 2. Biological characteristics of nematodes. After mating, nematodes of both sexes produce eggs and complete a developmental cycle through 3 periods: larvae, adults, and eggs. Normally, nematodes reproduce in 3 to 4 weeks, and several generations can occur in one growing season. The larvae of nematodes generally have 4 ages. The first, instar larvae, mostly develop in the eggs. The second, instar larvae, which hatch, can infest the host. The majority of plant pathogenic nematodes are living parasites. The parasitic mode can be divided into ectoparasitic and endoparasitic. The endoparasitic mode is when all the nematodes burrow into the plant tissues and only feed by piercing the host tissues with oral pins, while the nematodes remain outside the plant body. Different species of nematodes have different host ranges. Some are highly specialized and can only parasitize a few species of plants. Others have a wide range of hosts and can parasitize many different plants. Most of the nematodes live in the soil tillage layer, so nematode activity is not widespread. So, nematodes spread over long distances, mainly through the transport of seedlings. When feeding, nematodes pierce plant cells directly with their oral needles, stimulate cell division, forming tumors or excessive branching, inhibit division of the apical meristem of the rootstock, cause collapse of the mesocosm layer, and cell dissociation, rot. Most nematodes cause disease in the below-ground parts of plants, usually manifesting as weak dwarf, slow-growing plants with pale, even yellowing leaves. Major groups of plant pathogenic nematodes. 1. Cyst nematodes of the genus Cyst nematodes are heterozygous. Males are elongated. Mature females are lemon-shaped and pear-shaped. The cuticle becomes thicker and darker brown at maturity and are called cysts. 2. Root knot nematodes are similar to cyst nematodes and are also heterozygous. Mature females are pear-shaped. Males are long and slender with short tails. The main difference between the genus root knot nematodes and cyst nematodes is that the affected roots are swollen and form tuberculate root nodes, and the cuticle of the female does not change color after maturation. 3. The nematodes of this genus are hermaphroditic, long, and mostly parasitic on the above-ground parts of grasses, forming galls on stems and leaves. The genus grain nematodes includes at least 17 species. The type species is wheat grain nematode, which causes wheat grain nematode disease. 4. Stem nematodes. This genus of nematodes is hermaphroditic, linear in shape, and long in body. The nematodes of this genus affect above-ground stems, leaves, and underground roots, bulbs, and tubers, etc. Symptoms are mainly tissue necrosis and decay. 5. Slippery nematode genus. Hermaphroditic, linear with long oral needles, mainly affects plant leaves and shoots. Characteristics and diagnosis of nematode diseases. 1. Most of the symptoms are plant-wide: dwarf plants, yellowing of leaves, deformed and swollen roots or cysts in the roots. 2. Local symptoms are leaf blight, necrotic spots on the roots. 3. The nematodes can be isolated from the affected areas, such as root knots and seed galls. 4. Ectoparasitic nematodes need to be isolated from the soil and tested by artificial inoculation to determine their pathogenicity. The following is an example of root knot nematode to introduce the control methods of root knot nematode disease. 1. Crop rotation, preferably 2 to 3 years of crop rotation with grass crops. 2. Clean fields and cultivate disease-free seedlings. Remove diseased roots and destroy them centrally to reduce nematode density in the field. 3. Biological control. You can apply biological fungicides, such as pale purple cyanobacteria, to reduce the damage of nematodes. 4. Physical control. Flooding method to 5 to 30 centimeters soil layer for several months of silt irrigation can inhibit nematode infestation and reproduction. Solarization method. In the summer, high temperature season, every 10 days or so, deep plowing and turning the soil. A total of 2 uses of high temperature and drying to kill the nematodes on the soil surface. 5. Soil disinfection. Before planting, steam disinfection can be carried out during the recreational period, seedbed soil or shed soil, chemical disinfection before planting. 2. Disinfection at planting. 5% avermectin granules, 10% thiazolidine granules, 5% averthiazolidine granules, etc., can be applied in holes or furrows. 3. Chemical root irrigation can be used in the adult stage of the disease. Avermectin and thiazophos methyl can be used in accordance with the concentration of dilution and root irrigation. Thank you for watching.